Hello everyone, welcome to our dissection of the sheep kidney, and as mentioned previously, we'll only be talking about the details which are relevant for the anatomy physiology laboratory course NS 3420. I'm Dr. Michael.
I'm Eric, and I'm going to be walking you guys through the external anatomy of the kidney.
I'm Tantan, and I'll be walking you guys through the internal anatomy of the kidney.
And I'm Catherine, and I'll be walking you through the vasculature of our dyed specimen.
I hope you enjoy our video.
We will begin by examining the outer covering of the kidney. This is a connective tissue membrane called the renal capsule. It protects the kidney from trauma and other damage. Next, note that there is an indentation on the medial aspect of the kidney. This is called the renal hilum. In this area, the ureter exits. Also in this area, a large renal artery enters the kidney. This supplies blood to the organ. Also at the renal hilum, we see the renal vein and the ureter that leave the kidney. This is the vein, and this is the ureter. As we observe the walls and the caliber of each with a probe, we can observe that the renal artery has a much thicker tunica media, and that accounts for its rounder lumen. The vein has a much thinner tunica media, and that accounts for the more floppy lumen. The ureter has a distinct lighter colored tunica adventitia with the smallest lumen. The small size of the lumen is understandable because the volume of the urine passing through the ureters are much less than the volume of blood carried by the arteries and veins.
With the dissection knife, I've performed a frontal or coronal cut through your kidney specimen, beginning at the side opposite to the hilum, so this way. Please note that my fingers are above the blade. We can see that the outermost layer of the kidney has a slightly granular appearance. This is called the renal cortex. Deep to the cortex lies the renal medulla. This layer contains 8 to 12 triangular-shaped renal pyramids. Notice that the renal pyramids are cone-shaped, with the base lying adjacent to the renal cortex. That's the base. The apex, also called the papilla, is seen lying towards the medial aspect of the kidney. These are the papillae. Each pyramid is separated by a projection called the renal column.
So here we are going to appreciate both the urine collection and the vasculature of the kidney with our dyed specimen. When we can see here that our yellow structures represent our urine, our red structures represent our arteries, and our blue structures represent our veins. I'm first going to talk about the urine collection. Our urine is going to collect in the renal pyramids and then dump into our minor calyces. The minor calyces will then meet into a major calyx. The major calyces are then going to dump in to the renal pelvis. The renal pelvis will then dump the urine into our ureter, which can be appreciated here. Now going through our vasculature, our oxygenated blood is going to enter through our renal artery into our segmental artery, which can be appreciated here. Although we cannot observe the rest of the arteries in this kidney, I will still walk us through. The segmental arteries will then branch off into interlobar arteries. Those will then branch off into arcuate arteries. And we can appreciate here our cortical radiate arteries, cortical being cortex, and they are radiating outward. The cortical radiate arteries will then transport the blood to the glomerulus via the afferent arteriole. The blood will pass through the glomerulus, then will exit the glomerulus via the efferent arteriole. The blood will then either pass through the peritubular capillaries and or the vasa recta, depending on if you have cortical nephrons or juxtamedullary nephrons. The blood will then pass through the venules, which will then empty out into our cortical radiate veins, which you can see here. Our cortical radiate veins will then empty into our arcuate veins. Arcuate is another word for bowing, so you can see it bowing around. The blood will then empty out into the interlobar veins. It is important to note that kidneys do not have segmental veins, so that deoxygenated blood will then empty out into the renal vein, which can be appreciated here.